Hello, my name is Nicole Salz, and I will be presenting with Michelle Joyce, and we will be conducting a literature review on cortical folding in premature infants with cerebral palsy. Cerebral palsy, commonly known as CP, is the most common form of childhood disability, affecting close to 800,000 people in the United States. CP is typically caused by complications to the brain during the development period and impacts movement, muscle tone, and posture. Close to 50% of children who develop CP were born prematurely. This correlation is so high as preterm birth disrupts brain development due to the mechanical processes occurring in the forming brain, one of which is cortical folding.
For this project, we systematically reviewed 6 studies that focused on the connections between cerebral palsy, premature birth, and cortical folding. These studies obtained results by analyzing MRI scans of people born pre or full term and with or without cerebral palsy.
Many of our reviewed studies included comparisons of brain structure between preterm infants and full-term control infants. One of the main differences found was a reduced sulcal depth for preterm infants with cerebral palsy. This reduced sulcal depth relates to 3 of the main metrics that affect abnormal cortical folding.
The metrics we look at are gyrification, cortical surface area, and brain weight. People with cerebral palsy tend to have a decreased gyrification index and a reduced sulcal depth as compared to a typical folding pattern. As seen in the middle chart, there is also a decrease in cortical surface area in the preterm group compared to the full-term control group. This measure is related to the gyrification index and sulcal depth decreases. The cortex is also tended to be thicker and less complexly folded in this group. Finally, the histogram of brain weights shows that most of the cerebral palsy cases studied were about 50 to 80% of the weight of a normal brain at the respective age.
From these studies, we see that preterm infants have significantly affected cortical development. Due to the high percentage of individuals with CP born prematurely, we also see that these individuals exhibit great morphological variation. These deviations from the typical brain include decreased gyrification index, cortical surface area, and brain weight ratio. There are several hypothetical causes for abnormal cortical folding in development. However, the direct effect of cortical folding in children with CP is limited in both its data and research.
Although there is no specific known cause of cerebral palsy, we know that cortical folding is affected in individuals with CP and that premature birth is a risk factor. These changes in cortical folding patterns result in permanent neurological effects, but further research into understanding the direct causes of CP could help improve the diagnosis, treatment, and prevention. Additionally, ensuring proper care to reduce premature birth could reduce the prevalence of cerebral palsy. Thank you for watching our presentation.